Jai Hind everyone, I am Pankaj Kumar and this lecture is regarding the proteoglycan, glycoprotein and the glycolipid. So this is the series of the lecture and this is the third lecture of that series in which we are completing the carbohydrate structure. So if I will ask you about this very simple thing, you everybody know the antithrombin. So antithrombin, what is the function of the antithrombin? So it is the function will be the and it will prevent the conversion of the prothrombin to the thrombin. So like that. So it will prevent the blood clotings. So the same time, if you will find this clotting will be not occur in the so many of the peoples. So how the same person will get the more clotting faster and some person will not get the clotting faster and even some person will not get the clotting even at all. So this is the chapter which will give you the concept that how the protein which is present in you, like suppose FSH is there in me and you also, but among ourselves, some of the FSH will be more active in our body and some of the other person will have the less active. The same time, if you'll see the growth factors, so their functions, growth factor FSH or the any other hormones or any other proteins, glycoproteins, their functions will be defined on the surface of the membrane, and this will be all done by the proteoglycan and their interactions. So, what we are going to study this, and why we need to study this chapter, because this chapter will provide you the understanding about the communication between the cell and their extracellular surroundings. Second thing, it will give you the label proteins for transport and the localization in a specific organelle destruction of malformed protein. So the most third serve as the recognition site for the extracellular signaling molecule like in a growth factor or bacteria or the viruses like this. So let's begin with this basic understanding of the proteoglycan, glycoprotein and glycolipid. First we are going to see the basic meaning of the proteoglycan, glycoprotein and glycolipid. Proteoglycan, the macromolecule of the cell surface which are joined covalently with the protein. It can be either as a membranous proteoglycan or it can be as a secreted. Second is what? Glycoprotein, varying in the complexity, joined covalently to a protein found as a part of the glycocalyx and in blood found in a specific organelle such as a Golgi complex and form a lactin. So this is the important thing that it will form a lactin and it will help in the uh, blood group defining also. Now glycolipid is the plasma membrane component in which oligosaccharide is hydrophilic. So it is a plasma membrane component, as you know it will find in the glycolipid, and that glyco word will be hydrophilic in nature. So it will be basically on the head of the bilayer lipid. You can find in the neurons also. In neurons, the glycosphingolipid, which act as a nerve conduction, and it will help in the myelin formation as well as. So it will help in the signal conduction, signal transduction in the cell as well. Now we'll see the basic, not only basic, but majorly the proteoglycan. Proteoglycan. So proteoglycan are mostly the proteoglycan are the integral membrane protein, and some of them are in the basal lamina. What does it mean? It will be between the two group of the cells. Suppose there are the two cells, and there are the two cells, two group of the cells, and the basal lamella will be here somewhere. Okay, so basal lamella is between here. So this is the basal lamella in which extracellular matrix will be there. It consists of extracellular matrix, and this extracellular matrix contain the family of the protein, each with the several covalently attached heparan sulfate. So it will have a heparin sulfate, which we already studied in the last video. If you didn't watch the previous videos, first and second, you can click over here, the link which is showing here on the corner, and you can watch that videos. Now, heparin sulfate is what? It is bind with some proteins. Okay, so how it is going to be help in the cell interaction or the cell communication, we'll see. So basically, there will be two type of the families of this membrane heparin sulfate proteoglycan. Two families will be there. Membrane heparin sulfate proteoglycan. So first is what? The first is the Syndecan. So what is the important point about the Syndecan is, and why it is important? We'll see. So the Syndecan, it will be help in the single. It is a single transmembrane, single transmembrane, and an extra cellular domain bearing 5 to 3 chains of the heparin sulfate. In some cases, the chondroitin sulfate will be also there. So contain extracellular domain contain 3 to 5 chains of heparin sulfate, and in some cases chondroitin sulfate also. In some cases chondroitin sulfate will be also there, and mostly it will contain the heparin sulfate. So why this heparin sulfate will be very important for the cell signaling, that we'll understand. But first we'll see the glypicans. Okay, so the second families of the glycoprotein is what? Glypicans. Glypican is the attached to the membrane with the glycosyl phosphatidyl inositol anchor as a glycosylated derivative of the membrane lipid phosphatidyl inositol. So this is about the glycopine. So mostly the glycopican or the Syndecan, both will be the families which will be cut, like it will be trimmed or the cut over the inside the extracellular matrix only, and then it will become the part of the cell membrane. So as you can see here, sorry. So as you can see here, the protease will cut the protein in the extracellular matrix, and it will give you the Syndecan's extra ectodomain, and the phospholipase cut the connection to the GPI anchor, release the glypicans. So both will be cut by the protease or phospholipase, and it gives their respective. Like Syndecan will be cut by the protease, and the phospholipase will cut the uh, GPI anchor, and it will give the glypicans. So why this shedding or the cutting is highly regulated, and where it will be occur in the body? Can you guess why it is happening like this? Why this type of, why we need to cut again and again and give the syndicants? If there will be thousand different type of the cells in the body, do we have to cut different site, or we have to cut at the same time? Or syndecan and the glypicans will be same for the all the thousand protein cells? So you think, and you tell the answers in the chat box, in the comment box. But first see the Syndecan. Syndecan will be looks like this, in which you can find there is a, it is a single intermembrane protein, and it have a COH and NH3 chain. Now it will have the heparin sulfate, chondroitin sulfate, and the core protein. Now this core protein will be also same for the glypicans, but the glypicans, you can see this is a GPI anchor, glycosilyl glycosyl phosphatidylinositol. It is attached with the COH group, CO minus group, and it is forming a globular domain. So as you know, MSC molecule and the other molecules which are in glycosylated, that will be act as a glypicans. But in case of the syndicans, it is transmembrane protein, which is in the extracellular matrix as well as the inside the cell also. But GPI or glypicans is in the extracellular matrix. So how it will going to help the cells? Anybody can answer? It will act as a co-receptors like B80. Okay, in the immunoglobulin chapter we already studied. Okay, so co-receptors, it will act as a, glypicans will act as a co-receptors. Got it. Now the question will coming to the same question, and I am going to answer the question. Mostly the shedding will be occur in the proliferating cells. Okay, so cells which are not decided till it which function it have to do. Proliferating cells. So like if had to form the neutrophil, or if it have to be become a any B cells, so it will need a co-receptor, then it will form a glypican. If doesn't require a co-receptor, then it will form a syndicans, like that. So mostly this type of receptor will be uh, inhibited by the contact inhibition in case of the cancer cell. But this will be shedding or the cutting of this uh, glypicans and syndecan will be highly regulated in case of the cancer cell. Okay, so you remember it, in this cancer cell it will be not stopped, and it will be continuously cancer cell and differentiating cells. Okay, differentiating cell. Hope you understood till now. Now we'll see the next step, how it will be regulated, and how much the heparin sulfate is important for the cell signaling, or like for the for the cell communications and all. We'll see. The question is what? Now question is, how much heparin sulfate important for cell signaling. How much it is? So the answer will be something here. The answer is that the glycoproteins will be having a some important domains which will going to define the internal cell signaling and their communications. We'll see. First we'll see one diagram. So question is very important. Why heparin sulfate is very important for the cell signaling or their transmembrane protein formation, and how it will be proteins will be very specific for the particular proteins. So as you see, this is a heparin sulfate, and this heparin sulfate is same written here. Okay. So this heparin sulfate is made up of what? You remembered it. It is made up of the glycoproteins. Okay. Now this glycoprotein will have the several NS domain. What is this NS domain? This NS domain is nothing. It is just a non-random heparin sulfate domain. Okay. So non-random heparin sulfate domain consists of consist of it consists of around 3 to 8 disaccharides. Okay, 3 to 8 disaccharides. And this 3 to 8 disaccharide sequence is very important. Okay. So this 3 to 8 disaccharides long having a ability to bind with a specific protein. If it will be not there, then uh, it cannot be defined. Important thing is that this NS domain is non-random. It is not random. It have a some sequence, like after one sequence to alternate manner it will come like this. Okay. So this is very important. 2s is what? 2o sulfate, and 6s is 6o sulfate. Now iduronic acid and glucuronic acid and glucosaminoglycan. So you can see glucosaminoglycan, you can find it here. This is a glucosaminoglycan, and this is a NS domain. Now this is a one NS domain. This is the another NS domain. This is the another NS domain. So they are alternately alternately uh, arranged in a manner which will define the proper protein specific binding. Okay. So I'm going to write it the same statement, which is very important. The sulfated highly sulfated domain. So this heparin sulfate is very specific for the protein because it having a alternate sequence, and this is a it is a non-random sequence. Okay. So it is a highly sulfated domain like NS domain, and it is alternate with the having unmodified glucose N-acetyl glucosamine and glucuronic acid. So this is called as a Na domain. Okay. Glucuronic acid like uh, N-acetylated glucosamine and glucuronic acid will called as a anadomin. You can see in between this. Okay. So this is the anadomin, this is the anadomin, this is the anadomin, which is not exactly mentioned. It is just a line. So this is very important for the specificity for the binding with any protein. Now we'll see the heparin sulfate bind with the extracellular protein signaling molecule and alternatives. But why it had to be bind with the like alternative way? Like as if I will say here, suppose this is a NS domain, this is a NS domain, this is one another NS domain. Okay. And here one more. Now we have a what? We have a, suppose this is a ND domain, NA domain. Okay. Now why we need this alternate or a specific sequencing of a NA domain and NS domain? How it will help in the protein signaling molecules? So till now we know all the DNA sequence or the protein signaling will be on the DNA basis. Like DNA will decide which protein signaling will be formed, and that will go and bind to the particular cell, and it will give the XYZ type of signaling. But this heparin sulfate also modulate that signaling. How? We'll see. The first point is what? The first point question I should write so you'll get proper idea. So how it will uh, alter the protein signaling activities. So I want to show you one image, then we'll discuss it. So we can see the four cases according to heparin sulfate which will alter the protein activities. The first case is the conformational activation. Okay, so if the factor A will bind with the antithrombin will be activated and with the NS domain. Okay, so you can see a conformational change induced in the protein antithrombin binding a specific pentasaccharide NS domain allow its interaction with the blood clotting at 10th factor. Okay, preventing the clotting. Got it. This is easy and simple. If factor A 10 will bind with the antithrombin with the NS domain, then it will prevent the clotting inside the blood. Find it. So the change in the activity can be induced by the just by binding. Second case is what we can see. The heparin sulfate bind to different protein, bring them into close proximity and enhancing the protein-protein interaction. So you can see the enhanced protein-protein interaction by thrombin. So this is our again one antithrombin binding of antithrombin and thrombin to two adjacent NS domain. So antithrombin and this is one antithrombin and this is the thrombin. So this thrombin is binding with this domain and antithrombin is already bound with the one NS domain, and because of this close proximity that inhibit the blood clotting. Got it? This is easy only now. Now the third is what the heparin binding to growth factor enhance their interaction with the growth factor on the cell surface. Here heparin sulfate act as a co-receptor. So you can see uh, so heparin sulfate suppose like a Syndecan. Okay, you can write the heparin sulfate like a Syndecan. It bind with the growth factor. Okay, it bind with the fibroblast growth factor. This fibroblast growth factor and having a extracellular protein signaling. If the fibroblast growth factor will bind, the cell will go for the cell division. So heparin sulfate in the Syndecan will bind with the growth factor receptor. This is the important thing. What is the important thing? First, this FGF cannot directly bind with this protein, this FGF receptor. So what it is happening? This FGF is binding with this green color line. And what is this green color line? This green color line is the heparin sulfate. Now heparin sulfate will brings this FGF ligand to the FGF receptor dimer, and then cell will start for the cell division. They will go with the cell division. And now the most important thing is what? The cell surface localization and concentration. Whenever you will see some complementary components, so most of the complementary component or the soluble proteins, they will be concentrated with the heparin sulfate. So lipoprotein lipase high density negative charge in the heparin sulfate attract the positively charged what? Lipoproteins. If this positively charged lipoprotein lipase molecule will come to the heparin sulfate negatively charged and it will be concentrated here. Okay. The important thing is what? The NS domain interact electrostatically as well. Okay, it will interact electrostatically. Already mentioned here also, it will interact by electrostatic sequence specific interaction with the NS domain. The lipoprotein lipase will concentrated over the cell membrane. So how it is important and good to see how it is working. So that's why heparin sulfate is very important for the cell signaling and all. Okay. Important of the heparin sulfate domain. How to find? how we'll find the importance of the heparin sulfate? So is there any other function for the heparin sulfate? How much we have to read? Shit. So is there any other function for the heparin sulfate? Okay. Oh shit. So yes, we do have. We'll see. So what we can do? Make. Let us smile. So what we can do for the finding the importance of the heparin sulfate? Okay. Importance of heparin sulfate. To find it down, what you have to do? You have to do the simple thing. You have to do the simple thing. What it is? Just knock down. Okay. Knockout. So what is the meaning of knockout and knockdown? So knockdown is a down regulation of any gene expression, but knockout is elimination of a gene. Elimination of gene. So there is a one enzyme which is called there is one enzyme which is called iduronate 2 sulfatase. Okay. Okay. Iduronate 2 sulfatase. So what we are doing? We are doing the knockdown of this iduronate 2 sulfatase in the mouse. Okay. In mouse. So scientists found if this 2 sulfatase will be not there, then heparin sulfate will be not formed. Okay. If the heparin sulfate will not form, if this will not form, so they found the three important thing is happening. What is the first thing? The heparin bond, sorry, the mouse bond without without kidney, without kidney, abnormalities, abnormalities in skeleton and eyes. So if you find if there is a heparin sulfate lack of the heparin sulfate because of that enzyme, so the without kidney the mouse is born, and without kidney life cannot be expected, and there will be abnormalities in the skeleton as well as in the eye. These many things will be occurred. So other experiment also happened did by the other scientists, and they found this following results. If the heparin sulfate will be not there, so proteoglycan cannot help in the cleaning of the lipoprotein from the blood. Okay, so liver cannot cannot clean blood lipoprotein protein without proteoglycan. the last result. So s- evidence that proteoglycan contain the heparin sulfate and chondroitin sulfate provide directional cue for the axon growth. So it is already evident, evidence is there that proteoglycan direct axon growth. Basically, heparin sulfate we are talking about. So these are the very important thing. If you'll see, this type of the disorder will be occur in the bones. As you can see in the diagram, the bone deformities in this case. So whenever there is a defect in the enzyme or degradative enzymes, which will degrade which will degrade the glycosaminoglycan. So it is a defect in the degradative enzyme, means which are cleaning the glycosaminoglycan. So if this enzyme will be defected, then accumulation of there is a what? There is a accumulation of this glycosaminoglycan in the body. So if this is accumulated, then it can cause this disorder. It can cause CUET syndrome, which will which will affect as a joint stiffening and with the normal intelligence in the normal lifespan. But it can become a severe in case of the Hurler syndrome. If it will be more accumulated glycosaminoglycan, then it will cause dwarfism, intellectual disabilities, heart disorder, and as well as it can cause a early death. So this is the importance, like how much importance their enzyme degradative enzymes are there, and the proteoglycan activities, heparin sulfate are very important. So the third parameter to see or understand the proteoglycan is their proteoglycan aggregate. Okay, so proteoglycan aggregate. So what happen in the proteoglycan aggregate? The fibrous matrix proteins such as the collagen, elastin, and the fibronectin forming as a cross-linked meshwork that gives the whole extracellular matrix strength and resilience. So this all will help in the extracellular matrix strength and the resilience, which will be going to help collagen, elastin, and fibronectin. But the fibronectin is very important. It can bind with the fibrin and heparin sulfate and the collagen. The fibronectin and the other protein, which is present in the extracellular matrix, it will having a important sequence RGD. Okay, what is this RGD? Arginine, glycine, and asparagine. Okay, so arginine, glycine, and asparagine. It is in fibronectin. If it is present in the fibronectin, what will be occurring? So through which they will bind with the family protein called integrin. Because of this, it can bind with the integrin. So integrin will help in the signaling into the between the signaling help in signaling between extracellular matrix and cell. Okay, and between the cell. So this is all about the proteoglycan. Proteoglycan is finished over here. Now we'll see the glycoprotein and we'll see the glycolipid. So here is the glycoprotein. You can see the glycoprotein can be of two type. O-linked glycosylation will be there, or like in the N-linked glycosylation. So O-linked glycosylation usually will be seen in the serine and threonine, or an N-linked glycosylation will be seen in the asparagine. So here you can see this is bound with the O-linked, and here it is bound with the N nitrogen group. So glycosamine, N-acetyl glycosamine will bind with the serine, or N-acetyl glycosamine will bind with the asparagine. So if it is binding with the serine, it will bind with the oxygen. If it is binding with the asparagine, it is bind with the nitrogen. So like that, there will be mucus secreted, or it will be secreted, or the membranous protein will be there, or there will be different type of glycoprotein like immunoglobulin, FSH, or LH, or thyroid stimulating hormones. So most of the carbohydrate may constitute from 1% to 70% of the glycoprotein by mass. About half of all the protein of mammals are glycosylated. This is about the glycoprotein. Now we'll see the glycolipid. Glycolipid, most popular is the ganglioside. So ganglioside are the membranous lipid which will have the polar head group because of the carbohydrate. So the part of the lipid that form the outer surface membrane is a oligosaccharide contain a sialic acid. This is important. It will contain a sialic acid. But the gangliosides, like a glycosphingophospholipid, determine the blood group ABO. Blood group we can see in the different chapter in which we'll see how it will determine the ABO blood group. Like a glycolipid, like a lipopolysaccharide, which is present in the gram-negative bacteria, such like a Salmonella typhi or the E. coli, is the characteristic feature of the gram-negative bacteria. So thanks for watching till now. We'll meet in the next video. If you want to solve more questions, you can go and check into the description box. Some related questions will be available. And thanks for watching. We'll meet in the next video. Jai Hind, Jai Bhim.